Hey everybody, Mr. Grove here. Today we're going to talk about eukaryotic and prokaryotic cells. And so eukaryotic cells are going to be you. So that's how you can remember that. These are going to be plants, animals, protists, and fungus. And as we can see, they're very complex. There's a lot packed in there. They have membrane-bound organelles. So like the nucleus is very dominant and obvious, and that's going to be the eukaryotic cell. Prokaryotic cells, these are going to be more like bacteria and archaea, and they're very small. In fact, comparatively, they'd be about the size of this mitochondria here. And there's not much going on inside. They do have some things, but they're small and not near as complex as the eukaryotic cells, and they do not have a major nucleus that is obvious then. On the eukaryotic cells, there are three that we'll see, three structures that are kind of unique to the animals, and so that is lysosome, centriole pair, and flagella. And while not all animal cells have flagella, all animals do have species that have flagella. And so that is the lysosome, centriole pair, and flagella. Centriole pair is going to be used during cell division, and then the flagella, or the lysosome, is going to be used for digestive and kind of the breakdown of various things, so food particles, enzymes, things like that. The plants are going to have three things unique to them as well: cell wall, chloroplast, and central vacuole. And so those three items are going to be found in specifically plant cells. So the central vacuole we'll see is a water storage thing, and then the chloroplast for photosynthesis, and then the cell wall to give structure around the cell. So those are some things, eukaryotic versus prokaryotic. Let's go ahead and talk specifically on the prokaryotic. So prokaryotic cells, they do not have a nucleus, and like I said, this is bacteria and archaea, so they're pretty small in comparison. And so the first thing would be the nucleoid region. So this is where the DNA is going to be in. So where you have 46 DNA molecules in your nucleus, in this area, the bacteria cell is only going to have one DNA molecule, and it's just kind of a pile of spaghetti, kind of a Clark Griswold Christmas lights in here. And that contains all the genetic information for the bacteria for the most part. So that's the nucleoid region. Ribosomes are going to be these little dots, and they are sprinkled all throughout the cytoplasm. And that is going to be where proteins are made. So just like you make proteins, bacteria can also make proteins. Then these next three are going to be kind of layered. So capsule, cell wall, and the plasma membrane, or sometimes it's called the cell membrane. So a capsule is going to be the outside, and this is a sticky coating that helps move that bacteria from one location to another. So, when you touch a surface, that capsule might cause the bacteria to stick to your hands, and then hopefully for the bacteria, you would move it to a surface that's more suitable for its existence. The cell wall is always going to be the middle layer, and so that is going to be this rigid, tough structure that gives a little bit of protection to the cell, as also kind of the structure and rigidity for the cell. And then the cell membrane, plasma membrane, is going to be kind of like the bouncer. It lets some things in and lets some things out. And so it kind of controls what enters and exits the cell. The pili are these little short, spiky projections, and they are going to be like the capsule and help hopefully move that bacteria from one spot to another, not by doing something on its own, but by you sticking to it. So kind of like a cocklebur as you walk through the woods, and you move it from one location to another. And then the last thing is the flagella, and so often many types of bacteria would have flagella, and that would allow them to move. It's a tail, but tails are only going to be good if you have a water, and so that would help them move through a watery surface. So those are the features of the prokaryotic cell, and it's important to note that these are not considered organelles per se, but more just kind of components. As we switch over to the eukaryotic cell, these are organelles, and so these are the things that you have in your cell. And so the first is going to be the nucleus, and the nucleus is kind of the brains of the cell. This is where all the genetic information is going to be held, and it's what causes that cell to be that cell. So you have a skin cell and you have a hair cell. They both have all the genetic information, all 46 molecules of DNA. But what causes the skin cell to act like a skin cell and what causes the hair cell to act like a hair cell is the nucleus. So the nucleus kind of runs the show and causes the cell to act and produce the proteins and things that it does. Close to the nucleus is going to be the rough endoplasmic reticulum. And so that's kind of a mouthful, so we can just kind of refer to it as rough ER. It looks rough because under a very powerful microscope, we would see these little dots here. Okay, compared to smooth ER, there's not as many dots. And so it looks bumpy or rough underneath a microscope, and so that's why we call it rough ER. Those dots are indeed ribosomes. And so it's because of the ribosomes that it appears rough. And as we know from prokaryotic cells, ribosomes are used to build proteins. And so this is where the proteins of your cell are being built and manufactured. And so that's the rough ER. Compare that to the smooth ER. So it's not going to look rough, as we see here in our diagram. It doesn't have any dots. And so that's going to be the smooth ER. And the main thing the smooth ER would do would be the synthesis of lipids. So it's going to build lipids. So like your fats, your phospholipids, waxes, cholesterol, steroids, things like that. And that will be manufactured in the smooth ER. And both of these would be relatively close proximity to the nucleus because it's kind of like they're the factories and the nucleus would be the corporate headquarters. Moving on, we have one more manufacturing organelle, and that's the Golgi apparatus. And so it's kind of represented by these layers. It looks like it's kind of flat layers, lines. And these Golgi apparatus will receive proteins. So proteins will come in, and then they will tweak them out and customize them. Remember, a protein is very specific on its shape. And so then as it leaves, then it will be the perfect shape to be able to do its function. So it's kind of like, if you've ever had a key made at the hardware store, the rough ER makes that blank key the protein, but then the Golgi apparatus is like that fancy machine that then cuts all the little notches in the key, and then that ships it out so it can do its specific job. So Golgi apparatus means it's going to finish the proteins. Okay, and those are our manufacturing types of organelles. Now, the lysosome is going to be a very digestive organelle. So it's got these digestive enzymes. It's used to break stuff down. It's kind of like a little Pac-Man that's just kind of chomping through the cell. And so we can't let that happen. And so we have to section it off. So lysosomes are all oftentimes just kind of sectioned off in these little vesicles. And so if we want them to work on something, we will bring that something to that lysosome. So it could be food molecules, it could be an invader like a virus or a foreign cell, or it could just be an old damaged organelle that we just are ready to be done with, and so we're going to strip it down for parts and then use it to build up a new organelle. But that's the lysosome, and these are only found in animal cells, so not in plant cells. Vacuoles. So vacuoles are basically just storage compartments, little Rubbermaid tubs, and plants and animals will both have vacuoles because they both have to store things. However, a special kind of vacuole is only found in plants, and that's going to be the central vacuole. And it's this really large, obvious organelle. So if we look at a plant, that central vacuole is super huge in the middle, oftentimes bigger than even the nucleus. And so that central vacuole has one main job, and if you are a plant, the most important thing that you would have to store would be water. If you're an animal, water is important, but if you need more water, you just walk over to more water and get it from your supply. But if you're a plant, you're kind of limited to the water being able to come to you via rain. So you want to store as much of it as you can when you can, and that's the central vacuole's job. Often only found in plants then, for the central vacuole. Another plant only is the chloroplast. We'll talk a lot more about chloroplast next chapter when we deal with photosynthesis, but the main job of the chloroplast is photosynthesis. And so it's only found in plants, although there are very, very few exceptions like green hydra and some other protists that might have a little tiny bit of chloroplasts in them. So some chloroplasts they contain their own DNA and some of their own proteins, and so that we'll talk about that with mitochondria here in just a sec. But they're going to be green, which is because they have the chlorophyll. And so if this whole rectangle was a cell, these would all be the chloroplasts, all these little green circles. And so they're all throughout the cell, and their job is to produce photosynthesis for that plant cell. The mitochondria, this is what makes ATP, and the process it does that through is cellular respiration. So it's found in plant and animal cells. Sometimes we don't think of plant cells needing cellular respiration or ATP, but they do, and so they have mitochondria to do that. Kind of looks like a little jelly bean, and so that is the mitochondria, kind of thought of as the powerhouse of the cell. They also kind of have their own DNA, like the chloroplasts do. And so it's often thought of that they might have actually been on their own at one point in time and joined in with the rest of the cell as a symbiotic relationship at some point in time in history, because they have their own DNA. If you like the forensic shows, like CSI or NCIS, they often talk about mitochondrial DNA because it's very useful in isolating a particular individual organism. The cytoskeleton is like your skeleton, and it basically gives the cell structure, and it also gives a point of attachment for some of these organelles to kind of attach to. There are 3 different kinds of cytoskeleton, so those are microfilaments, and intermediate filaments, and microtubules. So a microtubule is kind of a hollow spring, so it's very flexible, whereas a microfilament is going to be more of a twisted fiber, and then an intermediate filament is going to be both of them, kind of the tubule around the filament. And so these give structure and support and shape to the cell, and allows, like we said, the organelles to attach or anchor to those points. Flagella and cilia. So these are going to be little tails. They're located on the outside of the cell for cells that do have them. And so they are usually made of these microtubules. So that's where that flexibility comes in. And so flagella are usually longer than cilia, and to be an effective tail, they kind of have to beat back and forth, and that allows them to move through the water. And so that's going to be the cilia and flagella. So, and then lastly, we have the cell walls. So cell walls are going to give that kind of outer covering support structure to plants. And so the thing is, if you're going to build a wall around your cell, it'd be like if you built a wall around your house, you'd be stuck in your house. You wouldn't ever be able to get something in or out. And so we have to have these little doorways, and these little openings are called plasmodesmata. And so that's the little doorway openings through the cell wall into the plant cell. So this page is a really handy little review that has all of those, and these slides are in your classroom notes, so if you want to pull up the slides individually. But that's the organelles of the cell, and I hope that was helpful.